Good morning, everyone. Welcome back to my channel, Molecular Mysteries. Hope everyone is doing fine out there. So, without any further delay, let's get into the class.
Come and fly away with me. Come and
Good morning, everyone. Welcome back to my channel, Molecular Mysteries. So, in the last class, we have discussed all about the prokaryotic cell and eukaryotic cell and all. And in this class, I want to discuss about the chromatin structure and function, which is the second topic of your syllabus to cover. Okay, so you should before I enter into the cell biology, like before I enter into the cell division and all the phases of cell division, you should be well aware of the chromatin and structure before you enter into the cell division. Okay, so I thought today let's finish the topic regarding the chromatin structure and function. So what is chromatin structure and function? So you know that prokaryotes and eukaryotes are mainly differentiated based on the cell membrane, nuclear membrane, and the nucleus. So if you take the eukaryotes, the cell contains nucleus and the nucleus mainly contains the chromatin-like structures. So what is chromatin? They are like fiber-like, thread-like structures which are present in the nucleus. So during cell division, these chromatin would get condensed to form a ribbon-like structure called chromosomes where the cell division happens. So this main chromatin contains the DNA called the genetic material. It carries the genetic material and hence it is very important for that to get condensed and distributed equally for your daughter cells. So today I want to talk about the chromatin structure and function. In the next class, I will detail explain about the chromosomes, their structure, types of chromosomes based on the presence of their centromere. Okay. So today's class, let's discuss about what is chromatin and what are the types of chromatin. Okay. So chromatin is a genetic material or the macromolecule that contains the DNA, RNA and the proteins. So the proteins that are present along with the DNA and RNA are the histone proteins. I will tell you in the next class like what are nucleosomes and how the DNA is wrapping those nucleosomes and what are the types of histone proteins. Okay. So they are called the chromatin structures. Okay. These are present in the nucleus of all the eukaryotes. So the chromatin mainly contains the genetic material which is important for you to travel for the generations. Okay. So the genetic material has the following functions. It prevents the DNA from getting damaged. It helps in tightly packing the DNA to fit into the cell, controlling the DNA replication and gene expression, and supports the DNA molecule for the cell cycling. Like you all know that cell has to undergo the mitosis and the meiosis, right? So okay. So till now you got to know what is chromatin, where is it present, and you may have some confusion before before tell you the importance like types of chromatin. Let me have a clear explanation about the differences between the chromatin and chromosome. As I told you, the chromatin is a composed form of nucleosomes, which are the complex of DNA and proteins. So nucleosomes are nothing but the histone proteins that have been wrapped by the DNA. Okay, they are called the nucleosomes. And what are the types of histone proteins? I will let you know in the next class. Okay, and the chromosome, I told you, they are the condensed form of chromatin. So when the chromatin, when they enter into the cell division, mainly like in the metaphase where you should be well aware of those also. So in the metaphase cell division, you will be seeing a ribbon shaped structure called chromosomes. So they are nothing but they are the condensed form of your chromatin. Okay. So chromatin is generally thin and long fiber, whereas chromosome, I told you, they look like a ribbon. They are like compact structures. Okay, this chromatin is found throughout the cell cycle, but this chromosome is mainly you can be easily seeing that in the metaphase where all the chromosomes will be place it in the middle of your cell cycle, like cell division. Pulled by your spindle fibers in the opposite cells. All the chromosomes are made to stand in the linear position, like in the center, right? So this I will discuss in the next class. As of now, please remember that this chromatin and chromosomes have got some differences. Okay. So What are the types of chromatin? Okay, for that I thought I can explain you better when I go for a diagram. Okay, see. So what are the types of chromatin? If you discuss, there are mainly about so these are the types of chromatin. If you are not comfortable with the red, I will go with the black. Okay, nice. See, euchromatin and heterochromatin. So you have already gone through the eukaryotes where you say U is a true. So right, if it is eukaryotes, why is it called eukaryotes? It is a true form of your karyon. Karyon means nucleus, and U is true form. So these eukaryotes contain the true form of nucleus, a well-developed nucleus. That is why they are called the eukaryotes. In the same way, euchromatin is called the true chromatin because they contain the genes that are genetically active. So what is genetically active? What does that mean? So for example, so in the last classes you have already gone through the replication process and transcription and translation where the DNA, if suppose this is your DNA, okay, this DNA for a DNA it has to undergo the transcription and produce an mRNA, right? This mRNA should undergo translation for the production of your polypeptide chain. So this is the process of your central dogma or whatever you say is basic thing in the cells that happen. But for the chromatin, what happens is, for example, this is your DNA, and that for this DNA to get transcribed into mRNA, there are some factors called as transcription factors. So you can go through the class that I have already gone through, and previously in my channel, go through for the transcription and translation, where you will come across the transcription factors. These transcription factors would bind onto your DNA, thus helping for your mRNA production and thus your polypeptide chain. So this is very important. That is nothing but called gene expression. If a DNA is able to express, it is called as gene expression. If a gene is able to express itself, it does mean that it's transcribed and it's translated. Okay, so every gene needs to be going through this transcription and translation. For this euchromatin, what happens is, like, suppose I want to say this is your nucleosome. It will wrap like this. Okay, I'm telling you, like, it is just wrapping all your here comes the difference. What does that mean? I just want to give you a diagram regarding this for you to understand. Like, see, okay. See, I'll tell you the difference. This is your linker DNA that links each and every nucleosome with one another. It wraps the protein, histone proteins, and it forms a nucleosome. See, when this DNA has to be transcribed, the transcription factors should come and attach here for your successful transcription to happen. That means the DNA should be first accessible for your transcription factors to go for transcription. If DNA is not accessible, then the transcriptions can't attach them. These transcription factors can't get attached to your linker DNA or to your DNA for your transcription. So for that to happen, transcription factors are to be there. But here there is no accessibility. So this is called the heterochromatin. So I will tell you what is the main difference between these two. So I told you in euchromatin, there is an accessibility. like you can say transcription factors can successfully attach to DNA for transcription and translation. But what happens in euchromatin? Heterochromatin. In heterochromatin, the name itself says hetero is like it's genetically inactive. Okay, like what does that mean? See, if you can understand, like, see, I'm again, I got a DNA here, but this DNA is not getting transcribed into no transcription and no translation. So there is no transfer or there is no binding of this transcription factor to the gene happening in heterochromatin. Why? Because there is no accessibility for the transcription factors to bind to the DNA. If you can closely notice here, you got the linker DNA space available between all the nucleosomes. But whereas in heterochromatin, you can see there is a tight binding of your histone proteins, so tight that there is no place for the transcription factors to attach to your DNA. Right? So, whatever the factors that are present can't be attached to your DNA, and hence the DNA can't be translated and transcribed in the process that normally happens. So, heterochromatin are commonly called inactive part of your chromatin. Okay, and euchromatin is the active part. So hope you got an idea like what are the differences between the euchromatin and heterochromatin. Also, not only based on this, they also differentiate these two chromatin based on the staining technique. Like this heterochromatin gives you a dark appearance because of high amount of nucleosomes attached so closely, and they are not loosely packed. Euchromatin is loosely packed, so it does stain a light color than the heterochromatin. So, like if you can, so hope you are clear with this. So, I will draw a small diagram here. What, how it really, you know, happens. Like which part of your chromosome does the euchromatin contains, and which part of it does the heterochromatin contains. Okay. So, for example, this is your I'm just drawing like this is your chromosome. Just it's my view, like I want to have a clear view. Okay. See, this is called a centromere. You know that this is called these are called chromatids. They are the halves of your chromosome. In the next class, I will tell you the shape and all. Okay. As of now, please remember they are called chromatids. And here, for example, this is your euchromatin. Euchromatin is arranged in this way. So when it is added with this state, it does look like a light colored. And in the same way, if you see the heterochromatin package, this is how your heterochromatin is packed in your chromosome. So it does look dark. So it is heterochromatin. Okay, so this is I have just drawn for your understanding. For a better understanding, I'll give you one more picture. Like if you can, sorry, like if you draw a diagram of your chromosome in detail. Okay, this is your centromere. See, these are called the telomere part. Okay, this is also your telomeres are the ends of the your chromosome. So what I want to tell you here is this part. Sorry. This is not quite visible. This part is called, like, this part is the place where euchromatin is present. So it is lightly stained. Okay, so this part is the chromatin where it is slightly stained. This part. This is where euchromatin is mostly present in your chromosome. Okay, so when you go for the heterochromatin, the darkest parts of your chromosome are the heterochromatin. These are the telomeres part and also the parts that are nearer to a centromere. These are all heterochromatic. These are all the darkly stained part. The blue stained part are all the euchromatin. See, this is your euchromatin. This is your euchromatin. This is your euchromatin. It is lightly stained. For your identification, I'm just drawing in a different color. Okay, and these are all the darkly stained heterochromatin. Okay, this is a euchromatin. They're generally dark in color. And this is your U. Okay, hope I am clear with the differences between these two. Right, so hope you got idea regarding this. I will also post the links regarding this for the better understanding. Okay, in the next class I will talk to you about the types of chromosomes, and based on the presence of your centromere, what are the shapes of chromosomes, and the number of chromosomes per cell, like what would be the deficiency if there are no proper number of cells expressed. Okay, I will all discuss that in the next class regarding the chromosomes. Okay, so see you in the next class and hope I am clear with the topic. So if you have any doubts, please let me know. I forgot to say one important difference also. Yeah, not the difference actually, like important slide where you come from the types of chromatin. So euchromatin and heterochromatin, the coil of chromonemata is not uniform all along the length of the chromosome. So chromonemata is one of the important structures of chromosome, where I will discuss in the next class. Okay, because of the uneven coiling of the chromosomes, they exhibit the different staining. I told you the staining substance of chromosome is acetocarmine. Okay, the regions with more coils are stained darkly, and such regions are called heterochromatin and generally inactive because they are not transcribed or translated. The regions with less number of coils are lightly coiled and they are called euchromatin, which are generally genetically active. So differential sustainability is called as heteropyknosis. Just please remember the term. Okay, just these are all the basic information regarding the chromatic. In the next class, I will deal with that in detail. Okay, thank you and have a nice day. See you in the next class. Until then, stay positive, stay healthy, and be kind to everyone. Thank you. Bye bye.